So, Unit 2 was over spectrophotometry. Essentially, what we were doing in this particular unit was we were using light to measure things. Okay? So as the name implies, metry, that means meter or measurement. Photo means light, and spectro means spectrum. So we're using the visible spectrum of light to measure the absorbance of various samples. So remember, with this visible spectrum of light, you're going to have varying wavelengths. Okay? Your violet, your blue, and to a degree your green are going to be at the lower end of the spectrum. Bless you. While your yellow, your orange, and your red are going to be at the longer wavelength end of the spectrum. Also remember, here the wavelength is short. So what does that mean about the energy? Is the energy low or is it high there? It's going to be high energy. Over here, I'm going to have a longer wavelength, but my energy is going to be lower, right? So your energy is lower when your wavelength is longer. Your energy is higher when your wavelength is shorter. If you ever get confused about that, think about the final two colors at either end of the spectrum, red and violet. There are two forms of light that we are exposed to on a day-to-day basis that have these colors in their names. Anybody here who picks up a television remote, you're working with infrared light. Infrared. Okay? Has anybody here heard of studies being done where television remotes have been linked to cancer? No, right? That's ridiculous. Okay? That's because infrared light has very, very long wavelength, which means low energy. Over here, though, with violet, what light are we exposed to on a day-to-day basis with this word in it? Ultraviolet, which of course has been linked closely to various forms of skin cancer. That's because it has higher levels of energy. Okay? Now, you guys went through and you defined ranges of wavelengths that correspond to each of these different colors. And I understand that the wavelengths you may have defined in those ranges might have differed between groups. What I would recommend you do, if you want to see how close you were to actually getting the appropriate range of wavelengths, is go into Unit 9, which is photosynthesis. Okay? You guys haven't done that yet, but if you go to the lab manual for photosynthesis, in one of the first pages of that lab unit, they're going to have the entire breakdown of the visible spectrum of light and the wavelengths that correspond to it. On the exam, you could potentially be asked to define the range of wavelengths that correspond to blue. Or you could just be given a random wavelength and asked, what color is this wavelength? Now with my students, and again this is me, so it doesn't necessarily apply to all instructors, if the wavelength I'm asking about is a borderline wavelength, so let's say that it's 480, which is approximately at the borderline between blue and green. Okay? If my students say blue or green at that point in time, at that borderline, I typically give them full credit. But not all instructors might do that. So again, study and see what your ranges look like compared to Unit 9's manual, and make sure that you're comfortable with those ranges of wavelengths, because your instructor, or I'm sorry, the people who write the practical could ask you anything pertaining to those, based either on what color is this wavelength, or what wavelength would correspond to this color. Okay. Questions, concerns about that? Rather than me just writing it, I'd much rather just let you go look at that table, because I feel that that way you have the information right in front of you and you can see how it correlates to what you guys got. Okay? Now, outside of this, we weren't just doing this in the chapter. We also were looking at the ability of the spectrophotometer as a device to measure the concentration of an unknown solution. All right, so let's go ahead and talk about that real quick. We're going to talk real quick about the spectrophotometer. So, depending upon I guess real quick, anybody here not go to this campus? Does anybody here go to Central or PRC? Okay, so a couple of people. All right, so I'm going to do my best to basically distinguish between the two different types of spectrophotometers we use here. In theory, most of them have similar kind of capabilities, but there are a couple of discrepancies. Okay? So, students who did lab here, you had your digital display for your spectrophotometer, and then underneath that you had a collection of different buttons that kind of did alternate things depending upon what you needed them to do. So you had one that was labeled lambda, like that. Okay. Then you had a series of buttons that included 0.00, and then you had like a little home button and the print button, which aren't really that important, but you know, whatever. And then finally you had this circle with these different like arrow-based buttons around it. Okay, now the reason I'm drawing all of this is because I want to go over a couple of core concepts regarding the spectrophotometer. First off, the 0 button. This is used to set the absorbance of the spec to 0. While this knob here was used to change wavelengths. Now, if you went to, yes, ma'am?
Can you explain what those buttons are again?
Sure, absolutely. This button here is the home button, and it's just like anything else, it takes you to the home menu. Okay? This button over here is the print button, which you guys didn't use. It's just used to print the screen. And then here, what you have is you have a circular button in the center, and then around it you've got 4 angular buttons that point in different directions. It looks like a compass rose. One points up, one points down, and the other 2 point in opposite directions. Okay? These buttons were typically just used to navigate through the menus. So again, not really that big of a deal. The buttons or knobs that I'm bringing to your attention, those are the ones that I consider to be kind of important. Because one changes your wavelength, and the other is used to set the spec's absorbance to 0. We call that zeroing the spec. Now, if you went to another campus, you had a slightly different kind of setup. At the other campus, you had a knob labeled wavelength. I bet you guys can't guess what that one does. And then beyond that, you had 2 other knobs, one that was labeled 0T, and the other that was labeled 100% T. Now, you're only going to be tested over the spectrophotometer that you use. So people from Spring Creek, you're only going to be tested over the one that we used here. People from other campuses, you're only going to be tested over the one that you guys use. The big thing here is the general idea of what the spectrophotometer is doing. Okay? So inside the spectrophotometer, we have a light bulb. And that light bulb, not surprisingly, is emitting light. You're going to take a sample of whatever you're measuring. You're going to place it into the spectrophotometer, and as the light hits that substance, some of the light is going to pass through it and go to a meter that's going to measure it and give us a readout. Okay, so here is our meter with our digital display, and it's going to give some form of readout in terms of absorbance. Okay? So, whatever light is trapped by the tube and the substance in that tube, that is the light that's being absorbed. Whatever light is passing through, that is the light that's being transmitted. So transmitted light is light that passes through the sample, and light that's absorbed is light that doesn't pass through the sample. It's trapped by whatever the substance is.
Would you mind just rephrasing that one more time?
Absolutely. So, light that is absorbed, when this light hits the sample, is light that does not pass through. Okay? So that light gets absorbed by the substance. Whatever light does pass through, we refer to that as the light that is transmitted by the sample. Okay. Right. So, this is the core concept. We're using absorbance and we're using transmittance with the spectrophotometer to tell us something. All right? Now, for those of you who came here to Spring Creek campus, essentially what you did when you placed your blank in here was you set the absorbance to 0, and we're going to talk about what that means. While those of you who went to Central Park and PRC, you probably looked instead at your percent transmittance first, and then your absorbance. So when we start talking about the blank, I'm going to talk about how absorbance and transmittance are related to each other in that regard, to kind of get you thinking about that. Beyond that, though, just in general, let's think about this relationship. So some light is absorbed and some light is transmitted. So if I have a solution that absorbs a lot of light, a ton of it, am I going to see a whole lot of light being transmitted? No. So how does my absorbance compare to my transmittance? If I have a really high absorbance, what am I going to see for my transmittance? Really, really low. Okay? But if that solution is terrible at absorbing light, such that the absorbance is really, really, really low, what can I expect to see for my transmittance? Really high. So everybody understands there's an inverse relationship between transmittance and absorbance. Awesome. Okay. So let's go ahead and talk about the overall goal that we had for this exercise. So your goal for spectrophotometry was that you had an unknown protein solution. So our goal was to find the concentration in milligrams per milliliter of an unknown protein solution. That's our goal. I'm going to box this in so we can just keep this in mind at all times. Now, I can take that unknown solution at any time during the lab and I can put it into the spectrophotometer. And that's all well and good. But the problem with that is that it doesn't give me the concentration in milligrams per milliliter. The only two values a spectrophotometer can give me are transmittance and absorbance, at least in the capacity of our lab. There are some more modern day specs that will give you concentration, but ours aren't one of them. Okay? So, I can get absorbance or transmittance, but I can't get concentration. So this poses a huge problem. I have no way to correlate concentration to absorbance and transmittance. So the cool thing about this lab was you guys actually acted almost like Rosetta Stone artists, where you found a way to bridge the gap between absorbance and transmittance and concentration. And the way that you accomplished that was by doing two things. The first was a serial dilution with protein standards, and the second was by creating a standard curve. So let's talk about the serial dilutions first. Serial meaning repeated, and dilutions meaning to make less concentrated. So you started with this particular sample called the protein standard that had a concentration of 10 milligrams per milliliter. So it starts with a concentration of 10 milligrams per milliliter. So what I could do with this is I could place this solution with a known concentration into my spectrophotometer, and let's say that when I did that, I got an absorbance of 0.5. Okay, so 0.5, and I know it's 10 milligrams per milliliter. Now if I take my unknown and I place it into the spectrophotometer, and it also gives me an absorbance of 0.5, what is its concentration? 10 milligrams per milliliter. Because I just took this one that I knew, and I got its absorbance. So does everybody understand how we're going to use this to help us figure out this? Okay, anybody confused on that particular aspect? Sir? Just still lost? Okay. All right, so think of it kind of like translating between languages. Okay? You've got a certain word that means something in another language. Okay? So for instance, I'll just take Spanish, not that I'm a native speaker, but I spoke for some time. Ropa in Spanish means what? Clothes, right? So anytime I see the word ropa in a sentence, I know that they mean clothes. Here, if I figure out that the absorbance is 0.5 of this solution, anytime I see 0.5, that tells me that the concentration is 10 milligrams per milliliter. Does that make more sense? Awesome. Okay. Now, what do you think the likelihood is that the lab staff was super duper nice to you guys and gave you an unknown solution that was 10 milligrams per milliliter? Not so much, because otherwise what's the whole point of the scientific process? Okay? So, as a result, we can't just take one data point. We need a multitude of data points. And that's where the serial dilution comes in. So the idea here is we're going to use this to dilute and give us more data points. Because the more data points we have, the more accurate we can be in our assessment of this unknown. So what you did was you took another 4 test tubes. And you placed 1 milliliter of water into each of those test tubes. Alright? So then, what you did was you took some of your stock solution, technically 1 milliliter of your stock solution, and you added it to the first test tube. Now, this solution is 10 milligrams per milliliter. Real quick, think about what these units are actually telling you. 10 milligrams per milliliter. What they're telling you is that for every 1 milliliter of that solution that you have 10 milligrams of protein. So if I'm adding 1 milliliter of this solution to this test tube, how many milligrams of protein am I actually adding? 10 milligrams, right? So I'm adding 10 milligrams of protein in that 1 milliliter. Now the person who said 5, you're jumping ahead. Like you're already chomping at the bit to do the calculation. Right? So I've added 1 milliliter to 1 milliliter of water. Let me ask you a real quick question. How many milligrams of protein are there in 1 milliliter of water? 0, right? Otherwise, what would the point of protein shakes be? You could just drink water and beef up however you wanted to at the gym, right? So, I've got 10 milligrams of protein, 0 milligrams of protein, and then I've got 2 milliliters total of solution. So my total amount of protein per milliliter here is technically 10 milligrams in 2 milliliters of solution. But if I simplify that, it comes out to how many milligrams per 1 milliliter? 5. So I've got 5 milligrams per milliliter in this tube. So what happened to my concentration as I went from the first tube to the second? It decreased by half. So then if I do the same thing here, and I pull 1 milliliter now out of this solution, which is 5 milligrams per milliliter, what is my new concentration going to be for this next tube? 2.5 milligrams per milliliter. Okay? And if I do the same thing here, add another milliliter, this time from this third tube into the fourth tube, what's my new concentration? 1.25 milligrams per milliliter. And then finally, if I take 1 milliliter out of that tube and place it into this final tube, my new concentration there is 0.625 milligrams per milliliter. Okay? Does anybody need me to go through why we got those different numbers? Everybody understand? Okay. Yes. So, essentially what I'm doing every time I go from one tube to the next is I'm diluting. And what that means is I'm putting half of the tube as water, which has no protein, and then I'm adding in a certain amount of protein. So example, again here, when I added 1 milliliter of solution, I was adding 10 milligrams of protein. But because I have a milliliter of water, I technically now in this tube total have 10 milligrams in 2 milliliters of solution. And when I simplify that, it gives me 5 milligrams per milliliter. Now if I take 1 milliliter out, I'm not taking out 10 milligrams, I'm only taking out 5. So I take 5 milligrams of protein out and I put it into this tube, which also had 1 ml of water, which has 0 milligrams of protein. So total in this tube, I have a volume of 2 milliliters, but I have 5 milligrams dispersed through that 2 milliliters, which means I have 5 over 2, which gives me 2.5 milligrams per milliliter. So every time I dilute, I go down in concentration by half. That work better? Okay, awesome. All right, so why did we do all of this? It's not because we're sadists or masochists. It's because we needed to get the concentration of the unknown. Each of these different concentrations is going to give us a data point on our standard curve graph. So when I finally graph everything, I'm going to have concentration on my x-axis, which is my independent variable. And again, this is in milligrams per milliliter. I'm going to have absorbance here, and every single one of these is going to give me a different absorbance. Okay? Now, real quick, before we get to this, at this point in time, if I place any of these tubes into the spectrophotometer, am I 100% sure that the only thing that has an effect on the absorbance is the protein in those tubes? What else do I have in those tubes aside from protein? I have water, and there was one other component you guys also added. What was it? Biuret. So, before I place these inside of my spectrophotometer and get these values, there's one other tube over here that I have to make. What was the name of that tube? The blank. Remember what the blank has. The blank has everything but the sample that you're interested in. So what we're interested in knowing is the protein and its absorbance on the light. We don't care about water, and we don't care about biuret. So our blank contains biuret and water. Now, for those of you here at Spring Creek, you made your blank, you put it into your spectrophotometer, and you pressed the 0 button. That set the absorbance of this tube to 0. What does that mean? Is this tube absorbing any of the light at that point in time? No. This is the equivalent to you placing your wayboat on the digital balance and zeroing that wayboat's mass out. Okay? Now those of you at the other campuses, you didn't press a 0 button. Instead what you did was you turned up the percent transmittance knob until the percent transmittance read 100. Now if you think about that, what that's telling you is that 100% of the light is passing through your sample. So if I've got 100% of the light passing through the sample, how much do I have being absorbed? 0. So no matter which way you did it, you did the exact same. Okay? So we finally get these different data points. They're not going to be in a nice straight line. And then what we do is we draw a line of best fit. We don't want to use the individual points to get our concentration of the unknown. We want to use a line of best fit. Because this linear representation is the best representation of all 5 of our data points. If you only look at one data point, you could be looking at an outlier. You don't want to do that. So then what you're going to do is you're going to get the absorbance of your unknown. Okay, so you're going to place this in the spectrophotometer, get your absorbance, and you're going to find out where that absorbance falls. So let's say it's 0.5. What I would do is I'd say, okay, my unknown is 0.5. So I'd mark it here on the y-axis, and then I'm going to come across. Now, a lot of students hit a data point, and then they come down. Don't be one of those students. Okay? You want to come across until you hit the line of best fit. And then you come down and you find out where that matches up on your concentration graph, or your concentration axis. And that tells you your approximate concentration of your unknown. So it's like reading a map old school. Okay? Any questions about that? All right. Let me go ahead and stop this real quick.